So let's talk about the cellular-mediated immunity system. Okay? And then after that, which we probably won't get to today, we'll talk about how the antibodies and the cellular-mediated systems work together. Okay? So thymic selection. We need to produce these T cells, but the T cells need to do what they're supposed to do. If you have a police academy, and I don't know how long it takes to become a police officer in the academy. Let's say 2 years. It might be 2 months. I have no idea. But let's say it's 2 years. You don't want to produce. You want to make sure you're going to produce good people and be able to. I'm not going to use the word shoot. You're going to be able to defend against the bad people, but you shouldn't touch the good people. Does that make sense? Okay? So you've got to make sure that they're trained well. You don't want to graduate in 2 months when it takes 2 years because they won't know what to do. So T cells mature in the thymus under what we call negative and positive selection pressures. So this is what's happening here when we use the police academy. We could negatively select them. In other words, if the police officers, after 2 years, they're just shooting good guys and bad guys, they can't decipher between the good guys and bad guys, we got to eliminate them. They're just not going to, we can't put them out in the field. It's just not going to happen. So they fail out. And then we phagocytize them and destroy them and use the parts for other things, right? And we use it for other things. We call this apoptosis. We destroy them and use the fragments for other things. We call this negative selection, all right? So this is where they're strongly against anti-cell. They can't really tell the difference between the two. Then we have positive selection. This could be two different things going on here. Again, using the police academy analogy. We can have some of the police officers where they fail to recognize self-antigens. So they just destroy the good guys. They don't recognize them. Not a good thing either. So we got to eliminate them. That'll be destroyed, again, apoptosis. But then there's other ones that the T cells, they do recognize self-antigens, or let's say cells, but they're very weak in doing that. Maybe they bind to the MHC, but it's very weak. They kind of break off. So these kind of police officers, they need remedial work. They can recognize it, but they got to build up their strength so that they can stick on them better. Does that make sense? So they need remedial work. So these are going to need to, they don't get destroyed, they're just going to go through the whole, maybe, summer school, so to say. All right? They're going to take a little bit longer to be selected. So this is just showing you again, negative, it's opposite the way I showed you, but here's negative selection, and here's your two positive selections. See, this one over here, it's very weak. It doesn't, is this the one? Yeah, this is very weak. So they're going to rekindle them a little bit. Okay? Questions on that? All right, a little bit of review on this. We're going to talk about them. Overview of T cells. Helper T cells, remember they have a CD4 on them. They're also known as T4 cells, TH cells. These will secrete cytokines and get the B cells and other T cells to that area. These are what I always consider probably the most important cells of the immune system. This is the dispatcher when the master calls for help. The master calls for help and there's no answer there, you're not going to get the SWAT team or the police officers or the fire department at that spot when the dog's barking and the burglar's there and whatnot. We have the cytotoxic T cells, also known as killer T cells, same thing. And these have a CD8 on them. They're also referred to as T8 cells. And these will destroy viral infected cells and also destroy foreign human cells, meaning like for when you get a kidney transplant. We also have suppressor T cells, and they do also have the CD8, but we don't refer to them as the T8 cells because it'll be confusing when we talk about the killer T cells. These are going to inhibit the B cells and T cells so that they don't work too much. So when we have someone who has an allergy, they might have a problem with their suppressor T cells because the suppressor T cells are not doing their job. You need to make sure that the police officers are not getting too haywire, that they're not gung-ho and they want to just shoot everybody around there. They need something to suppress them. And then you have the memory T cells, which again remembers that particular antigen from before. And again, the immune response is much faster and much greater on the second and all the other exposures from there on. Okay? So, helper T cells. These are the most abundant T cells that we have in our body, 65%. They help activate T8 cells to become activated killer T cells. They help activate the antibody-mediated system. They help activate B cells and plasma cells. They're going to produce interleukin-2 that's going to stimulate basically all the immune responses. And it's going to produce a clone of memory T cells, T4 cells, helper T cells, so that we can have long periods of time where we can, it'll last forever, okay? Again, this is our dispatcher, the helper T cell. We're going to talk about AIDS because HIV destroys the helper T cell. There's also something called a T cell antigen receptor, and this is actually the part that binds the antigen, binds to the antigen presenting cell over here. All right, so the APC releases the interleukin-1 that we talked about before. All right, but this is the part that attaches to the antigen and presents it to the antigen presenting cell. Okay, or the T cell. T4 cells release cytokines, and we talked about these interferons. That's when the dog is barking and alerting all the neighboring houses that there's a burglar in the neighborhood. Right? We talked about that. So interferons interfere with viral replication in other cells, and it also activates natural killer cells, NK cells. We also have something else that gets released, which is going to be the macrophage activating factor, which enhances the physiology of macrophages and stimulates macrophages to release interferons. And it also releases interleukin-2, which is basically going to stimulate all the T cells and B cells for that matter. Okay? So, again, interferon, the MAF, and interleukin-2. Okay? So let me just put it all together for you. This is a nice little summary of what we've been talking about. You can put your pencils down. This is important, but everything I'm going to explain is written down here, but it makes it very much easier for you to understand. Here's an antigen-presenting cell, a macrophage. It sees something here, a bacteria, and there's antigens around it. This macrophage is going to engulf this bacteria, break it up with a lysosome, and a little piece of it goes out. And attaches to a protein that this macrophage made. The protein is an MHC. And in this case, it's MHC class 2. MHC class 2 has to bind to what type of T cell? 2 times what is 8? T4. So this presents, as an antigen-presenting cell, it's going to present to the acquired immune system. In this case, the helper T cell. The helper T cell sees that, makes a connection. A CD4 comes out and makes it more sturdy. And when it does, makes a connection here, this macrophage is going to release interleukin 1 to activate this. When this gets activated, the helper T cells, and the helper T cell is going to release interleukin 2 and all those things we just talked about. Interleukin 2 is going to activate killer T cells, is going to activate B cells to make antibodies, and it's going to activate this cell to go through mitosis and make more helper T cells. Notice if you destroy this, you don't get any of this. And that's what HIV will destroy. This is that person calling up the dispatcher. Does that make sense? Can you visualize it? In my videos that my lecture videos, I'm also putting in other videos I found on YouTube, and I put them in there too. Short things, like 2-3 minute ones. Okay? And this is showing you all the different things. There's our helper T cell that goes to the MHC class 2, and of that little mushy thing that's up there I can't point to, but that's the antigen presenting cell, or macrophage. Once the helper T cell gets activated, it releases interferons, it releases MAF, interleukin 2, interleukin 2, interleukin 2, and it does all these things, which I'm not going to go through with you again, but that's what they do. It's a good summary of what the helper T cell does. So last time we were spending more time discussing helper T cells, and like I said before, helper T cells I consider probably the most important cell in the immune system. A burglar comes in and breaks through the picket fence. Fido, the dog, starts barking. Again, the picket fence and the dog are non-specific portion of the immune system. The dog's barking is going to alert the master, who's very specific and realizes, okay, Fido, or whoever the dog is, that's the mailman. It's okay. It's friend. You can have fun with him. It's fine. The burglar's coming in. Again, the master identifies the burglar and says, that's a foe. That's an enemy. So now he's going to call up 911, the dispatcher. Okay? So he's becoming more specific. When he talks to the dispatcher is what I consider in this analogy the helper T cell. Because that helper T cell, that dispatcher, is going to call whatever services need to come to that house. If someone had a heart attack, the ambulance would come there. If the burglar's there, the police officers would be there. If there's a fire, the fire department gets there. The dispatcher is very, very specific and very important to get everyone at that area. Does that make sense? That's the helper T cell. And when you get into HIV, that helper T cell is what gets destroyed. Imagine yourself calling for the 911, and there's no answer. It just rings and rings and rings. That burglar, that fire, or that person who had a heart attack is going to get worse and worse and worse because it can't get help through that helper T cell. That's why we call it a helper T cell. All right, does that make sense? Now, the people coming over, let's say the police and stuff, that's what this is. These are the killer T cells. The dispatcher isn't destroying anything. They're just going to get help to that location. Does that make sense? And that's what this is. The police are going to be the killer T cells or cytotoxic T cells or T cells that will actually do, I don't say damage, but you know what I'm saying, that they're actually going to see what's going on and destroy what they need to do. Is that clear? It's very specific. All right? So here's our cytotoxic T cells, also known as the T8 cells and the killer T cells. They make up about 35% of all our T cells. Okay? Which is a big amount, but helper T cells are bigger. Okay? They're going to recognize tumor cells associated with viruses, okay? There are certain cancer cells that are caused by viruses, and the one that's getting a lot of advertisement, which we've known, doctors have known for decades, but now we've only seen it on advertisements recently in the past few years, is HPV, right? Human papilloma virus, which causes cervical cancer. So it is a virus that causes cervical cancer. Hepatitis B can cause liver cancer. HIV can cause a certain kind of blood vessel cancer called Kaposi sarcoma. So there are some viruses that will cause cancer, and that's what these do over here. So killer T cells will recognize those tumor cells or cancer cells that are due to viruses. They'll also deal with somatic cells that, with viruses inside them. Okay, so these are more specific towards the viruses. Also, it's going to deal with the foreign transplantation cells. You get a kidney from your next door neighbor. Yes, it's a foreign thing to your own body, and it's going to, your immune system is going to attack that. Now, that might not sound good, but it means that your immune system is working appropriately, right? And it also will recognize bacteria, fungi, and tuberculosis, TB, okay, which we'll talk about when we get into respiratory. And they're going to lyse things. Lyse means to destroy, to blow up, okay? And it does require the co-stimulation that we talked about before with MHC class 1 and the killer T cell. Remember, class 1 times 8 will give you 8, right? My little mnemonic to help you with that. So you got to have that, and it's got to have some kind of cytokines, especially interleukin 2, that's going to activate the killer T cells, okay? And the interleukin 2 comes from these helper T cells, or T4 cells, either one. All right, so what's going to happen is this killer T cell doesn't get destroyed itself, all right? We don't expect the police officers to go to the house as a burglar, destroy the burglar, and destroy himself or herself altogether, all right? The police officer will go to this house, do what he needs to do, go to the next house, go do what he needs to do, and so forth. So what's going to happen is the killer T cell is going to have direct contact to that cell, destroy that cell, and then detach off that cell unharmed. It will be unharmed, the killer T cell, and then it will go and attack again to another cell. All right, so we can reuse this thing. It's going to secrete a few things. There's things called perforins, and we kind of talked about those before. Perforins get released from the killer T cells, and these are going to perforate. That's why they call them perforins. They're going to perforate the cell that has a virus inside of it and put holes in it. Right? When you perforate something, you put holes in it. That's going to allow the extracellular fluid to enter the intracellular fluid and change the membrane potential. And it ultimately will destroy that cell, and that's what's going to happen then.
When a virus or other infectious agent infects a cell, some of the proteins of the infectious agent are broken down into peptide fragments. These fragments are complexed with special proteins called Class I MHCs, major histocompatibility complex proteins, and displayed on the surface of the infected cell. The T cell receptor on the cytotoxic T cells interacts with the virus-infected cells by recognizing both the foreign protein fragment and the Class I MHC. The cytotoxic T cell then releases toxic substances which cause death of the infected cell. Cytotoxic T cells also produce a toxin called perforin, which creates holes in the cell membrane, causing the cell to break open.
It also releases lymphotoxins, which help kill enzymes inside the invader cell, so it won't be able to work properly. And it will also release gamma interferons. And if you remember what interferons are, the little burglar comes inside, breaks into the house, tiptoes as he did that. The dog is sleeping, and he breaks into the house, steals the jewels or whatever, leaves the house, and slams that gate. And that awakes the dog, and the dog is now going to bark and alert neighboring houses can't do anything to the house that it was protecting because it was already done, but the dog barking is going to tell other houses that there is a burglar in the neighborhood, so we need to make sure that it doesn't go into those other cells or into the other houses. So it's going to, basically the interferons are going to interfere with viral replication in other cells. That's what's happening there, okay? Okay, so let's look at this. All right, everything I just talked about is right here. You put your pencils down, and it'll also be written over here too. But this is what's happening. Here you have an infected cell that's infected with a virus. Okay, so I'm going to put a piece of it over here with a class 1 MHC. This is a killer T cell. There's going to be recognition over here, and it's going to bind here with a CD8. All right? Co-stimulation occurs, and what happens here is that when this killer T cell gets activated, it's going to release. These little purple dots are going to get released, and they're called perforins. They get released, and they enter the cell here, and it's going to allow extracellular fluid to enter the cell here and destroy that cell. Okay? It is your cell, but it's infected. We got to get rid of it. No problem. There's other cells that are surrounding that we can actually do mitosis and fill in those gaps. Okay? Questions about how killer T cells do? They do what they say. They're going to kill something. As much as the helper T cells are going to help the system. All right? But the helper T cells don't kill. This one kills. All right? So, you've seen this picture before, so let's put the whole thing together. Okay? Here's a cell that's been infected by a virus. The cell is going to take that virus and be able to break it up. Okay? And a little piece of it will come over here. We can get back to that over here. Meanwhile, the cell is still producing viruses. It's going to produce a lot of viruses. It has a slow response to destroy the viruses inside. So in the meantime, hundreds of viruses are going to spill out. And we explain that where there's going to be an incorporation of the viral RNA or DNA into the host DNA, and it makes copies. That's how viruses work. So you have this little virus that comes out of this cell and comes this way. The macrophage, which is an antigen-presenting cell, is going to engulf that virus, break it up in here, put a piece of it out here, and this macrophage is going to present that antigen to the acquired immune system. So this is an antigen-presenting cell. It puts that piece out by a class 2 MHC. The only thing that binds to a class 2 MHC is a CD4. 2 times 4 equals 8 by mnemonic. So here's a class 2. It binds with a CD4. This here is a helper T cell, right? A T4 cell. Once you've got co-stimulation over here and recognition, the macrophage is going to release interleukin-1 to activate that helper T cell. That helper T cell, once activated, is then going to release a number of different interleukins. One of the most significant ones is interleukin-2. All right? Remember, hot T-bone state, it's going to activate T cells. It's going to activate the killer T cell. This killer T cell, meanwhile, this cell over here did make a little, break up a piece of the viral end over here and put it here. It puts it on a class 1 MHC. The class 1 binds to a CD8, 8 times 1 is 8, right, by mnemonic. Once this happens, this killer T cell is then going to release perforins to put holes into this cell, allowing the extracellular fluid, which has a lot of salts in it, is going to change the membrane potential of that cell and destroy it. Meanwhile, helper T cell is also going to activate B cells. And B cells will turn to plasma cells, which will then be factories to make antibodies. And we talked about all the different functions of antibodies. Meanwhile, the helper T cell is also going to stimulate itself to go through mitosis and make more helper T cells. More dispatchers if there's a lot of problems going on in that town. And that's what's happening here. It's a very busy picture. It's a very confusing subject. And my way of teaching it was to teach you each one of these individuals, these players, and now you see how they all come together. In my little lecture recorded, or my recorded lectures that I've been doing for you guys, I've incorporated videos that I found on YouTube, short ones, 2-3 minute videos. Watch it, because you get to see how, this is just a picture, two-dimensional thing, but when they show the little videos, it makes more sense when you see the animation going on. I find that students have a tougher time with this because they can't see this stuff. It's like chemistry, you can't see it. Heart, you can see, you can visualize where the pressure is getting higher or lower. All right? Blood vessels, you can see where they're going. You get the digestive system, you can see that the stomach is related to the small intestines and so forth. This, you have to visualize everything. You got to almost rely on these animations to help you understand it. Okay?
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Any questions about how the whole thing interplays? And we didn't know all about HIV is what really started the whole thing, for us to gain our knowledge. Yes.
It's just where the infected cell is, and I understand that a piece of the virus comes out, but when you look over towards the macrophage, there's a circle.
That's a virus, and that's a piece of the virus. So what's happening here, this virus-infected cell is producing viruses. In the meantime, it's also trying to break up one of the viruses. So it's doing two things at the same time. Does that make sense?
But the virus is coming out of the cell, right?
Correct.
I thought it wouldn't come out of the cell, it stays in there until...
No, it makes the virus, it makes a copy of the virus, and the virus can go outside. That's how all viruses kind of work. They use, they're parasites. They rely on a host cell to use its mechanics so that it can make copies of the virus itself, and make hundreds of these. It's only showing one, but there's hundreds of these coming out. This happens faster than the cell breaking up one of the viruses. So there's going to be a delay for the killer T cell to come here. This is going to be producing a lot more viruses. So we have to rely on memory T cells, memory B cells, so that if there's another virus, that this delay has been shorter. But if it's the first time that that cell has ever, or that body has ever seen that virus, it's going to take some time. Like the chickenpox, it takes about 2 to 3 weeks for your body to actually respond to that. But then after that, you could be exposed to chickenpox. You don't see any manifestation on your skin, unless you have some sort of immune compromise, HIV, stress, just getting older. It doesn't come up as chickenpox, but it comes up as shingles. Does that make more sense? Okay. Anybody have any? This is I expect there's going to be questions on this. So if there is any, let me know, because this is a more difficult subject matter to handle.
There are two major classes of lymphocytes involved with specific defenses: B cells and T cells. Immature T cells are produced in the bone marrow, but they subsequently migrate to the thymus, where they mature and develop the ability to recognize specific antigens. T cells are responsible for cell-mediated immunity. B cells, which mature in the bone marrow, are responsible for antibody-mediated immunity. The cell-mediated response begins when a pathogen is engulfed by an antigen-presenting cell, in this case, a macrophage. After the microbe is broken down by lysosomal enzymes, antigenic fragments are displayed with MHC molecules on the surface of the macrophage. T cells recognize the combination of the MHC molecule and an antigenic fragment, and are activated to multiply rapidly into an army of specialized T cells. One member of this army is the cytotoxic T cell. Cytotoxic T cells recognize and destroy foreign cells in tissues or virus-infected cells. Another T cell is the memory cytotoxic T lymphocyte, which remains in reserve in the body. If sometime in the future these T cells re-encounter this specific antigen, they will rapidly differentiate into cytotoxic T cells, providing a speedy and effective defense. Helper T cells coordinate specific and non-specific defenses, in large part by releasing chemicals that stimulate T cell and B cell growth and differentiation. Suppressor T cells inhibit the immune response so that it ends when the infection has been controlled, whereas the number of helper T cells increases almost at once. The number of suppressor T cells increases slowly, allowing time for an effective first response.
Okay? All right. So keep in mind that if this is a phagocytic cell, it doesn't just do one thing at a time. There's going to be multiple things that cell's going to do. It's got to be multitasking. got, you have a receptor for bacteria, receptors for complement system, receptor for antibodies. You're going to have, and it's not just one, and one. Remember, they're just showing you this little funny diagram, but there's a receptor for bacteria all over the cell. There's this receptor for complement all over the cell. It's not just one receptor per cell. So this compounds the immune system astronomically. Okay? All right, suppressor T cells. This also has a CD8 on it, but we don't refer to it as T8 cells. That would confuse matters. T8 cells are only referred to in terms of the killer or the cytotoxic T cells. Okay, but this does have a CD8 on it. And it's going to suppress, as the name implies. It's going to down-regulate the immune response so that it doesn't get haywire. All right? You want it to, if you're going to have, let's say, a pollen going into your nose, you want your body to react to it, but you don't want it to react all over the place. Right? So that you can't breathe. So in most people, the suppressor cells do their job. It does hone down the immune response. In certain people, the suppressor T cells might not be working. So yes, there is a correlation with people who have allergies to pollen and their suppressor T cells might not be working properly. And that the immune system is hyperactive. Is that clear about that? All right. We don't know much about it, but with allergies coming up, because there's got to be some sort of interesting phenomenon happening. When I was a kid, people having peanut allergies, I guess one out of, I don't know, maybe I have one friend that had it. But now, peanut allergies, you go to the cafeteria in, let's say, 5th grade, they have a whole table where there's people with peanut allergies. You can't bring things with peanuts into school. I mean, it gets pretty bad. So there's some kind of phenomenon, I don't want to say phenomenon, there's something in maybe the foods that we're eating, something in the health that we're doing, I don't know. But there is going to be more and more about this. And if you're going into the health field, understanding the immune system is going to help you to understand what's happening with society, let alone your own kids, if your own child's going to be going through this. And then we have the memory T cell, which I think is self-explanatory. It just remembers that particular virus so that when the second time happens where your body gets exposed to that virus, we know exactly what to do, and it happens much faster. Okay? And this is just showing you, pretty straightforward, a lymphocyte. If it has a CD4, it gets activated and these can become helper T cells. And this, for the lymphocyte, if it has a CD8, it becomes a killer T cell or a suppressor cell. All right? And the activation or the maturation of T cells occurs in the thymus. Right? That's why we call them T cells. And there's a certain hormone that gets released by the thymus to mature these lymphocytes to become T cells. And what is that hormone called? Thymosin, right? Okay? And that's what that is. B cells get mature in the bone marrow, and you don't see that up here, right? But the lymphocytes do start off in the bone marrow, but they get mature if they're going to become a T cell in the thymus.